Hi everyone, it's Dr. LeBlanc with a video capture lecture on DNA replication in prokaryotes. The learning goals for this lecture are to learn the three phases of a DNA polymerization reaction: initiation, elongation, and termination, specifically in E. coli, but these principles apply to many other organisms as well, including eukaryotes. We are going to learn the components of the replisome and all their individual functions. We're going to understand the features and mechanisms of DNA polymerase itself. We're going to understand the differences between leading and lagging strands of DNA synthesis and the 5' 3' polarity of the template and the newly synthesized strands. We're going to look at one mechanism of termination, and then we're going to learn the biochemistry of the polymerization reaction itself. So DNA replication, as does all polymerization reaction, has three stages: initiation, elongation, and termination. Usually, initiation is the most complex of all three of these phases of any polymerization reaction. A bacterial chromosome is a circular chromosome, and it has a specific origin of replication, nucleotides of a specific sequence to which the proteins of the replication machinery bind. Once DNA replication is initiated at the origin of replication, it proceeds around the circle to create two replication forks. This is often called the theta structure because it resembles the Greek letter, theta. These forks proceed around the circular structure until they reach the termination site here, and then the two new E. coli chromosomes separate from each other. If you radioactively label the nucleotides that are the precursors to DNA synthesis, you can use a photographic emulsion to detect that DNA structure. And this is exactly what this is. This is an actual autoradiograph of an E. coli chromosome in the act of replicating. And this is the interpretation here. So that this theta structure here is resembled this theta structure up here. So if we want to diagram our origin of replication and how DNA synthesis is initiated at it, we have a sequence of nucleotides here at the origin of replication and two replication forks. This is a bidirectional DNA replication event where we have a single leading strand here on this side from this fork and multiple short fragments on the lagging strand. And the reciprocal is seen on the other side of the replication bubble in this replication fork. This is the actual DNA sequence of the origin of replication on the E. coli chromosome. Notice there are some features that we can annotate. There are five different DNA A boxes. A box, in this case, is a sequence of nucleotides that has a function, usually to bind a particular protein. In this case, it's the DNA A protein. DNA A binds here, 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 and here. So there are 5 different molecules of DNA A which will bind to the DNA sequence here. Notice that they are next to some AT-rich regions that are also part of the origin of replication. What do you think the function of the AT-rich regions might be? Well, an AT base pair has only 2 hydrogen bonds compared to a GC base pair, which has 3. So a region of the DNA that is AT-rich requires less input of energy to separate the 2 strands than would a region that was GC-rich. Here is a diagram of our double-stranded DNA with the DNA A boxes and the AT-rich region. The first event that occurs during the initiation of DNA replication is the binding of the DNA A protein to these individual boxes. The DNA A protein not only binds to the DNA, but additional proteins also bind to the DNA and to each other, causing the DNA to bend sharply in this region. When the DNA bends here, the AT-rich region pops open. And once that initial separation has occurred, it allows other proteins of the replication machinery to come in and attach to the single-stranded DNAs at the forks and proceed with DNA replication. The first protein to come in is DNA helicase, and it travels along the DNA in both directions. The DNA helicase separates the DNA, creating two replication forks. The bidirectional replication. Let's look at DNA polymerase III, which is the holoenzyme from E. coli. DNA polymerase III is the DNA polymerase of the replication machinery. There are three replication DNA polymerase in E. coli, 1, 2, and 3. We will see a specific role for DNA polymerase I in just a minute, but polymerase III is the replication DNA polymerase, and it has many subunits. It has an alpha subunit, which actually has the catalytic activity in it. It has an epsilon subunit, which contains a 3' to 5' proofreading activity, which is very important for a DNA polymerase to have. It has a theta subunit, which is an accessory protein that stimulates the proofreading function, and it has the beta clamp, which we talked about previously. The clamp protein allows the DNA polymerase to slide along the DNA without falling off. The beta clamp is what makes DNA polymerase highly processive. Then it has a whole other complex of proteins that is the clamp loader complex, which is involved with helping the clamp bind to and come off of the DNA. This is very important, especially in the lagging strand of DNA synthesis. This is a diagram of DNA polymerase III. The holoenzyme is an asymmetric dimer. Here's the clamp over here. The clamp loader. This is the clamp on the either side. This is the clamp loader. So this is going to be very important for the DNA polymerase on the lagging strand. Okay, this is the nitty-gritty of the actual DNA synthesis reaction. Okay, I'm going to tell you something very important about all DNA polymerases. All DNA polymerases can only extend a hydrogen-bonded nucleotide with a free 3' hydroxyl. Okay, I'm going to say that again. DNA polymerases can extend the 3' free hydroxyl of a nucleotide that is hydrogen bonded to a template. It cannot initiate DNA synthesis on a single-stranded DNA template. It needs to extend a nucleotide with a free 3' hydroxyl that is hydrogen bonded to the template. Okay, that is true for all DNA polymerases. Okay, so what is the reaction that we see here? This hydroxyl attacks this phosphate diester bond from an incoming nucleotide triphosphate. This is the building block of the DNA molecule. It has a base, a sugar, and 3 phosphates attached to it. These 3 phosphates fuel this reaction. They provide the energy for this synthesis reaction. So here's the new ester bond here, right here. Okay, the oxygen from the phosphate is now attached to the 3' carbon of the previous sugar, the end of the molecule that we're extending. Okay, so this is the new bond that's formed right here. When this bond is broken and pyrophosphate is relieved, the pyrophosphate will then further hydrolyze and yield additional energy for this particular reaction. So the DNA synthesis reaction doesn't need additional ATP. The energy to fuel this reaction comes from the nucleotide triphosphates that come in and add to the growing chain. Now we have a new 3' end that is one nucleotide longer than our previous strand of DNA. This is a model of a DNA polymerase. If you hold your right hand up in front of the screen with the fingers pointed up and slightly to the left, or down and slightly to the left, your thumb up in the air and your fingers curl up, your hand will be another model of DNA polymerase. Here's our DNA, the newly synthesized DNA in the light blue, and our template DNA here. So our template is being fed into the DNA polymerase, and the new nucleotides are being added at the catalytic site. The incoming deoxyribonucleoside triphosphates come in here and get added at this point. Notice there is also a 3' exonuclease proofreading site. This is extremely important for the accuracy of the DNA synthesis reaction. Here's a space-filling model of our DNA polymerase. Okay, here's another model, a ribbon diagram of DNA polymerase. Here's our newly synthesized DNA, double-stranded DNA. Here is our single-stranded template coming in. This is the catalytic site down here, and up here is our proofreading site. Notice they both have molecules of magnesium, which are very important for the catalysis, either the synthetic reaction or the exonuclease reaction. If there is a mismatch, the wrong nucleotide is added to the growing 3' end. The change in the conformation of the DNA causes the catalytic activity to pause. The DNA in this conformation has a lower affinity at the catalytic site and a greater affinity at the 3' exonuclease site, the proofreading site. The 3' end of the newly synthesized strand of DNA enters this exonuclease site, and the nucleotides are removed one at a time until the bad nucleotide is excised. Once you remove the bad nucleotide, the conformation of the double helix now is preserved, and it has a lesser affinity for this site and a greater affinity for the catalytic site, and the DNA moves back to the catalytic site, and nucleotides are started to be added again one at a time. If DNA polymerase did not have this activity, they would be error-prone, highly error-prone. And when you insert the wrong nucleotide into a DNA sequence, you create mutations. And for the most part, mutations are deleterious. So over the course of evolution, DNA polymerases have evolved to have not only the DNA synthesis catalytic site, but also the exonuclease site that removes bad nucleotides. Different DNA polymerases are better at proofreading than others. Okay, now we're going to look at the replication fork. We're going to look at the role that primers play on both the leading and lagging strands. When you separate the two strands of the template, one of the strands is in the 5' end, has its 5' end at the beginning, and the other strand has its 3' end at the beginning. But DNA replication can only proceed in a 5' to 3' direction. This top template strand then allows DNA synthesis to proceed uninhibited through the entire length of the DNA molecule. Once you prime the DNA synthesis with an RNA primer, DNA polymerization can occur uninterrupted as the replication fork proceeds down the DNA molecule. However, on the opposite strand, there's a problem or an issue that has to be dealt with. Once a segment of the DNA is separated at the initiation of DNA replication, and a primer is laid down, DNA replication occurs from here out towards the end. But we need to replicate this DNA as well. So when more DNA is separated, a new primer is laid down by a specific protein, and another short fragment of DNA is synthesized. These short fragments are called Okazaki fragments, after Mr. Okazaki, and they are a way to replicate this lagging strand of the DNA molecule. As you proceed down the length of the DNA, you add a third Okazaki fragment. Well, what happens to those first two Okazaki fragments? They are ligated together after the RNA primer has been removed. And that process continues all the way down the length of the chromosome. The newly synthesized Okazaki fragment is ligated to the previous Okazaki fragment that is a part of this now long DNA molecule. What creates those primers? It is this protein right here. It's called DNA-G or primase. DNA-G is an RNA polymerase. RNA polymerase, unlike DNA polymerase, can synthesize a new nucleotide without having to add it to a previously hydrogen bonded nucleotide on the template. So here's a diagram of DNA synthesis with almost all of the players. We have the leading strand here that is synthesized continuously and our lagging strand on the bottom. We'll start with DNA helicase here, that is breaking the hydrogen bonds. Once those hydrogen bonds are broken, in order to keep the two strands of DNA template separated from each other, single-stranded binding proteins come in and bind to keep those two strands of DNA separated. Here's our topoisomerase or gyrase that is relieving the stress of the coiling that occurs when you separate the two strands. DNA polymerase 3 is the replicase, and here it is on the leading strand of DNA, adding one nucleotide after another uninterrupted. On the lagging strand, the DNA polymerase has to be loaded on and taken off and loaded on and taken off as it extends new primers laid down by the primase. Remember, this is an RNA polymerase, so the primers here indicated in red are all RNAs. The Okazaki fragments are ligated together by DNA ligase. Now there's one important player that's missing from this diagram, and that is the enzyme that extends the end of the newly synthesized Okazaki fragment and removes the RNA primers. That enzyme is DNA polymerase 1. It has not only a 5' to 3' DNA synthesis activity, but it also has a 5' to 3' exonuclease activity that allows it to chew up the RNA primers. Once it chews up the RNA primer, however, there's still a gap right here that DNA ligase has to seal up. So here are a whole list of proteins that are involved in DNA replication. There are most of them, not all of them. A few of the important players are the DNA A protein, the helicase, topoisomerase, single-stranded binding protein, primase, Pol3, Pol1, ligase, and to terminate DNA replication, we need a TUS protein. So here's a replisome with all the important players, and we can go through and make sure we understand all of the players and what they do. DNA helicase, the topoisomerase, the primase. Together, the primase and the helicase are known as the primosome. They're single-stranded binding proteins. There's an RNA primer here. Don't forget DNA polymerase 3, the featured performer here. Here's a new Okazaki fragment. Here's an older Okazaki fragment. And here is DNA polymerase 1 right here that extends the end, the 3' end of the new Okazaki fragment, and it chews up the RNA primer to leaves the gap that DNA ligase will then seal up. Here is a very, very excellent interactive animation website that you should definitely check out, and a list of a few others that are also very good. Termination of replication occurs when the fork reaches the TER sites. There are some proteins that help to separate the two strands, two intertwined strands of the two new chromosomes. They're called the TUS proteins, and they bind at the TER sequences on the DNA. These proteins allow the advancement of the clockwise moving fork and prevents the moving of the counterclockwise moving fork. This sequence right here with the protein bound to it, and the same is true for the opposite. This allows the advancement of the counterclockwise moving fork and prevents the moving of the clockwise moving fork so that they don't run into each other. At the very end of replication, after the two chromosomes are completely replicated, they are actually linked like magician's rings. They're catenanes, and you have to use a topo 2 to break those two chromosomes apart so that one chromosome can go to one new bacterial cell and the other chromosome can go to another bacterial cell. Here is our summary slide. There are 3 phases of DNA replication: initiation, elongation, and termination. Bacterial chromosomes are circular. They often have 2 replication forks, allowing for bidirectional synthesis. DNA synthesis always proceeds in a 5' to 3' direction, resulting in a leading and lagging strand at each fork as DNA is copied from both parental strands. Initiation begins at an AT-rich origins of replication. DNA polymerases are multi-subunit proteins, each subunit having different specific functions. DNA polymerases all need a primer with a free 3' hydroxyl. During replication, the primer is usually RNA. The synthesis reaction adds a new nucleotide to the free 3' hydroxyl using the energy from the nucleotide triphosphate. DNA polymerases also have proofreading and exonuclease activity. There are several different DNA polymerases with specific roles in the cell: DNA polymerase 1, DNA polymerase 2, and DNA polymerase 3. Lagging strands are synthesized in shorter fragments called Okazaki fragments that are linked together by ligase. The replication machinery consists of many components with specialized functions. They are, and you should be able to list them: termination of circular chromosomes occurs at specialized TER sites. Linked chromosomes are resolved by topoisomerases.